Thanks for the introduction, Julian. Can I just check if everyone can hear me before I get started?
Yes, yep. Can hear and see you perfectly. Thank you so much.
Great, thank you. So today I want to talk to you about the various cellular molecular mechanisms that ensure neurogenesis persists throughout life in mice. And one of the reasons why we're interested in studying this is because it's been suggested, at least recently, that perhaps neurogenesis ends in humans once we reach adolescence. And so we're interested in identifying why it persists in mice, and perhaps identifying whether these mechanisms are present or absent from the human brain. So just a quick background on the hippocampal neural stem cell niche. So at the base of the neurogenic lineage is, of course, the quiescent neural stem cell. So quiescence is a reversible cell cycle exit. These quiescent neural stem cells generate neurons when required, and these neurons have important functions throughout adult life. Because these neurons continue to be added all the way through to end of life, in a sense, the dentate gyrus is continually developing. Now, one thing that's really important to mention here is that the levels of hippocampal neurogenesis change with time in a pattern that's highly conserved between species, but poorly understood. And so here's just a quick schematic showing the hippocampus with the dentate gyrus, which houses the stem cells here in this V-like structure. And the neural stem cells are here on the left, and at any given time, the vast majority of these, about 95% of them, are quiescent, and only about 5% of them are actively dividing. When they divide, they generate trans-amplifying cells, which over a period of 4 weeks generate mature granular neurons. So what is the pattern of hippocampal neurogenesis that I alluded to? So the pattern of hippocampal neurogenesis is that it occurs in highest levels in the juveniles of any given species. And while neurogenesis is at its highest in juveniles, this is also a period in which it is declining at its greatest rate. Then something happens sort of after sexual maturity where this rapid decline stops, and instead there's prolonged maintenance of low levels of neurogenesis all the way through death. And this can be seen in the schematic here on the right. So on the x-axis, you have age in days, and the y-axis, you have dentate gyrus neurogenesis expressed as a percentage of its maximum value. And this is a rodent hippocampal neurogenesis. And as you can see in the first sort of 2 months of life, neurogenesis decreases from 100% of its maximum to about 10%. And yet a similar tenfold decrease takes the rest of the mouse or rat's life. So clearly at this sort of period of sexual maturity, something happens to ensure neurogenesis continues, albeit at lower levels. And as I mentioned recently, or at least 2 years ago now, a paper came out in Nature suggesting that perhaps this process or this transition to low levels of neurogenesis doesn't occur in humans. Instead, the process just terminates, which perhaps is not good news if you think that ongoing neurogenesis is a good thing. So the first hypothesis we wanted to test was a fairly simple one. Seeing as neural stem cells form the base of the neurogenic lineage, presumably this change in the production of neurons has something to do with a change in stem cell behavior itself. But the problem we faced was there's different models about how stem cells behave in the hippocampus. They all seem to contradict each other. And they all seem to be published in the same year, 2011, in the same journal. This first paper, which is a favorite of mine, described stem cells behaving in a disposable manner, where the quiescent neural stem cell will activate, enter the cell cycle, and divide 2 or 3 times in a consecutive manner without returning to quiescence, and then deplete through differentiation. And the net effect of this is that the stem cell pool shrinks. And as I said, in the same year, another group came up with a completely opposing model where they suggested that when a quiescent neural stem cell activates, the cell will divide and generate neurons, but then the cell usually goes back to quiescence and self-renews for prolonged periods, up to a year. So these models are very different from one another. They're chalk and cheese. More recently, our group suggested a sort of heterogeneous model, which is very similar to the last model, but there's one sort of key difference I want to highlight. So here we basically give the quiescent cells 2 different names. So we call quiescent cells that have not before divided since the establishment of the niche, we call them dormant cells. And when these dormant cells activate, these active cells can then generate neurons, but occasionally they will return back to quiescence. And the difference with our model is that we call these cells that have returned back to quiescence resting cells. And this is because we think that for some reason, they have different physiological properties to those cells that have not divided before. So this leads me to the second hypothesis of the talk, which is that perhaps this transition is caused by stem cells switching between these various models. So early in life, where there's a rapid decline in neurogenesis and high levels of neurons, stem cells are rapidly dividing, generating neurons and disappearing. But with time, they switch to a long-term self-renewal mode. And so it's not that these models are incorrect or entirely correct on their own. It's just that they're correct at different stages of life. And so that's what we wanted to investigate in this project. And so the first thing we tested was to see whether stem cells first behave according to this division coupled depletion model, or the disposable model, which it's sometimes referred to. And the nice thing about this model is it makes a really testable prediction. So here it predicts that the level of stem cell proliferation or activity should match the rate in which these stem cells are disappearing. And what we found was that in young mice, the predicted and observed values matched, but after sort of sexual maturity, for some reason, proliferating stem cells become resistant to the process of differentiation and depletion. So why is this? Well, we hypothesize that it could be because these cells might acquire the capacity to go back to quiescence. And perhaps this is why they no longer stressed out by proliferation and differentiate. And so to test this, we gave mice EDU for various periods of time. And then after a short chase period, we looked to see whether these proliferating stem cells were still proliferating, or whether they had gone back to quiescence and lost the cell cycle marker expressions such as Ki67 or MCM2. And this is just an example of a resting cell. So at the beginning of the experiment, the cell was dividing, so it took up the EDU label. But by the end of the experiment, it's now quiescent, so it's resting. And yes, what we found was quite a remarkable change, where that in young mice, stem cells never go back to quiescence, but they gradually and substantially acquire this capacity with age, so that by 6 months of age, the majority of cells go back to quiescence after dividing into this resting state. So I just want to quickly highlight some other cellular mechanisms that work to preserve this lifelong stem cell population that we identified. So in addition to these cells going back to quiescence, we found that the bulk of the stem cell population, the dormant cells, progress into even deeper quiescence. So much so that they kind of force themselves out of contributing to homeostatic neurogenesis and end up being protected from proliferative stress. And because of the first and second effect, the net effect is that these resting cells, cells that have recently gone back to quiescence, end up being the predominant contributor to stem cell proliferation through time. So initially, dormant cells are the primary contributor to stem cell proliferation, but over time, this switches to this resting stem cell population. And so to build upon and to validate these findings, we performed single cell RNA sequencing of these different populations. So I don't have time to talk about the particular mouse model, but it's suffice to say, all stem cells in this mouse model are green, and then the cells that are proliferating and have gone back to quiescence have TD tomato fluorescence. And we sequenced cells at 1, 2, and 6 months of age. In our combined data set, we sequenced 24,000 cells using the 10x genomics platform, but only about 3,000 of these are bona fide quiescent neural stem cells. The rest are just cells that are labeled by the transgene. As many people will know who work in neurodevelopment, there's no one specific transgene that labels stem cells in an entirely specific way. And so once we had this pure population of quiescent neural stem cell and proliferating neural stem cell transcriptomes, we ordered these cells according to the most deeply quiescent cell to the least quiescent cell, using a tool called Slingshot to form a pseudo-time progression. And as you can see, cells that exist in the deep quiescent state in blue have high expressions of genes associated with lipid metabolism, whereas genes that are active are expressing genes that respond to growth factor signaling, for example. So this is just a validation of our ordering approach. And consistent with our immunohistochemistry experiments, we found that the bulk of the stem cell population in a dormant state are deeply quiescent, whereas these aqua cells, these resting cells, exist in lower dimensional space much closer to these proliferating cells, indicating they're in a lighter state of quiescence, as our immunohistochemistry data suggested. Likewise, when you look at their transcriptional profile, you see that they express, well, even though they are still quiescent, they express lower levels of quiescent genes compared to these deeply quiescent dormant cells. And on the other hand, they express genes associated with biosynthesis, such as RNA export and ribosomal biogenesis at higher levels than dormant cells, suggesting they're primed to enter the cell cycle when needed. Finally, we also profiled these dormant cells. So as I mentioned earlier in the talk, our immunohistochemistry data suggested that these cells progress into a deeper state of quiescence to sort of protect themselves from proliferative stress to preserve the stem cell population. And so when we look at these pseudo-time ordered cells and group these cells according to the age of the mouse that we analyzed, we can quite clearly see that the dormant cells that came from older mice, the 6 to 8 month old group here in magenta, existed in deeper states of quiescence compared to the dormant cells from 2 month old mice in orange. And then the cells from 1 month old mice were those that typically were closest to activating at any given time. So just to quickly summarize the cellular mechanism, and then I'll wrap up. In juvenile mice, most stem cells exist at the very tip of the activation profile. They're just ready to kind of fall off the cliff and proliferate. And because of this, many of them do. And when these cells proliferate, they generate neurons, and then the stem cell disappears. So you get the stem cell pool shrinking very rapidly. But as mice get older, what happens is that it is at least 2 things, or there's actually 2 or 3 different things happening at once. The bulk of the stem cell population, the dormant cells, go back to a really deep state of quiescence. So they essentially never proliferate, preserving their number, perhaps until they're required in case there's a tissue injury. But then the few cells that do activate, they acquire this capacity to go back to this resting state where these cells remain primed to activate. So you get this sort of partitioning of the stem cells into 2 different populations, and that helps preserve the proliferative potential while also maintaining the stem cell pool. And I don't have time to talk about the molecular mechanisms, but just to briefly summarize them, what we found was that in juvenile mice, this behavior is governed by high levels of AUSbiosearch protein. So AUSbiosearch is a transcription factor that acts as an activation signal, essentially, promoting the expression of cell cycle genes, for instance. But as mice get older, this protein gets degraded, not at a transcriptional, this protein gets downregulated, not through transcriptional downregulation, but through the activity of a ubiquitin ligase called HUV1. And we validated these results by looking at mice that had artificially lowered levels of AUSbiosearch, so not knockouts, but hypermorphs. And we performed immunohistochemistry studies and also single cell RNA sequencing of their transcriptomes to see that they resemble cells from older wild type mice, therefore demonstrating that this phenotype is caused, at least in part, by the reduction in AUSbiosearch protein levels. So just quickly, I'd like to thank my boss, Francois, the CRIC, and the science technology platforms that operate there. And that's a picture of us in Sicily last year in happier times. Thank you for your attention. and happy to answer any questions that you might have.
Wonderful, Lachlan. Thank you very much for your presentation. It's really remarkable stuff. I'm looking at the chat box, and we have some nice comments, and I'm welcoming questions from the audience. If I may start with one. Lachlan, that's really remarkable. And in the interpretation of your results, you explained that over time, these animals have stem cells that deepen in their dormancy. So you essentially interpret it to mean that there is not an extinguishment of their capacity to awaken from their dormancy, but that they do perhaps have some capacity, and so it's a quietening, not an extinguishment. So is there a way that these cells may be reawakened? So for example, your experiments with artificially reduced ASCOL1 protein show that these cells behave like a mature quiescent stem cells in a mature brain. But on the other hand, if you reconstitute, if it's too difficult to do, reconstitute ASCOL expression within the stem cells of the mature adult, would you expect? Or if you reduce one expression, for example.
Yeah, I think I would predict that those cells would then activate. They might take longer, but that's one hypothesis we want to test. So is that deepening of the quiescence of those dormant cells, is that adaptive? So does it actually serve a purpose? So perhaps the reason is the mouse wants to set those sides of cells in case there's an injury, for instance. So perhaps if we do some sort of injury model, those cells specifically will be recruited. So yeah, we'd be interested in trying to validate our speculation that perhaps that process is adaptive. It's not actually a bad thing for those cells to deepen their quiescence.
Thank you, Lachlan. I see that I have had similar brainwaves with Lara over east here in Australia. So Lara, do you have an additional comment that you might want to add to this particular point? If I can ask you to unmute, that would be great. Thank you.
Sorry, I forget. Is there any neurological conditions where you would maybe consider where that deepening of quiescence isn't adaptive and becomes more maladaptive? And maybe that reactivation of cells becomes less efficient maybe as you get older, that awakening, I guess, cells quiescence becomes less and less easy to do, and it would be I guess adaptive to come in and maybe artificially quote unquote reawaken them, I guess.
Yeah, it's a really great question. So other groups in our field have suggested that this process is pathological, but we kind of take the argument that we're still looking at very young mice. So our old mice are only 6 months of age, and it doesn't make sense. And it's actually these changes we see occur just after sexual maturity. So we're kind of making the argument that it's probably adaptive, but you're exactly right. Once these mice get to 12 to 18 months, 24 months of age, I imagine this process of deepening quiescence might become completely irreversible and senescence, and that's pathological. It's just for technical reasons in our field, people tend to use quite young mice and argue that they represent aging, but yeah, we're not really convinced by that because they're still very young, and we think that at least 6 months of age that perhaps is serving an adaptive function, but the question of 18-month-old mice or something like that is a different question altogether. Yeah, great question.
Great, thank you, Lara. I'm pleased to see that there are additional questions from the audience. I'll start with a question from Reese Knowles, and then Jerry has a question. So Reese's question is the following. I was wondering how and when you are inducing the mice with EDU. Also, how long do you think the EDU will label dividing cells for? And I can open the floor to Reese if he wants to follow up any comments from that. Thank you, Lachlan.
Yeah, that's a great question, and one that we got hammered for. So we've repeated, so actually I haven't updated the slide. So we repeated this experiment with a short injection of EDU, including to 18-month-old mice. And then that way we could be very sure of the birth dating, and to make sure that there wasn't any sort of, we didn't design the experiment. I didn't design the experiment very well the first time, but yeah, so a very short injection of EDU, and then we look 48 hours later to see if they lose Chi67. But we also repeated the experiment recently 1 week later to see they lose the expression of another protein called MCM2. Now, MCM2 is expressed for at least 4 to 5 days after a cell exits the cell cycle because it's super stable. So that way we could validate that these cells were returning to a bona fide quiescence and not just an extended G1, which is a technical argument some people were making to suggest that these cells weren't really returning to a bona fide quiescence. And so that was a hard one to address, but we think these resting cells do return to a genuine quiescent state.
Thank you, Reese, for your questions. Excellent. Jerry, you have a question. Thank you.
Yeah, cool. Great talk, Lachlan. So I was wondering whether, like, what exactly are the signals that are pulling those kind of active versus resting quiescent cells out of quiescence, and like whether the HUE1 and the ASCL1 interaction is kind of like a master regulator of that transition. Is there another? Are there other signals at play there, or is this kind of the pathway that you guys are proposing to be the one?
Yeah. So yeah, I'll take the second part first. So the ASCL1 is the master regulator. So if you knock it out, there's actually no stem cell proliferation whatsoever. So that was work published by a previous postdoc in our group. But yeah, we're certainly, so at the moment we're trying to dissect what are the downstream factors, and certain genes like the MYC family we think are responsible for later stages of activation. But yeah, ASCL1 is a master regulator. Yeah, as to whether resting cells receive different signals from dormant cells or whether it's completely like stochastic, not sure. But the resting cells don't seem to like position themselves in discrete locations within the dentate gyrus. They seem to be just scattered randomly. So that would perhaps argue that it's more just stochastic and as to which cell adopts which fate. But generally, when we do RNA velocity on the single cell RNA sequencing data, you see that all cells are kind of being pushed back into deeper quiescence. It's just probably random chance that some get activated and they can enter this resting state. And yeah, but we need to do more work to determine that.
Cool. And then I guess as a follow-up to that, is this also something you see in the SVZ, like the other neurogenic niche, and is it also something that you see in other stem cell niches that are perhaps a bit more active than this one?
Yeah, so people haven't looked at this dormant resting before in the subventricular zone, but certainly another group recently showed also that in the subventricular zone, with age, these cells do deepen their quiescence. So it seems to be a conserved feature, which is really cool.
Cool.
Thank you, Jerry, for your questions. And we have Emmanuel. I open the floor to you for your question. Thank you.
Yes, thank you. So my question is in relation with, for example, other animals, like mostly like lower vertebrates that can still, or they have more cell proliferations until later ages. If you think that these models could also be explaining some of these differences, and if through evolution, some of these mechanisms have changed in mammals and in higher vertebrates to change this proliferation rate, and if you could maybe study it also in lower vertebrates to see how this has been changing.
That's a fantastic question. And yeah, the only answer I have to that is that not many people study adult neurogenesis in other species. I mean, it's a bit of work in zebrafish, but for the reasons you outline, more should be done, because that could help address some of the mechanisms that we're interested in. So yeah.
Thank you, Lachlan. I'm going to interject with one more question, if I may. There has been, since Arturo Alvarez-Bulla's description of very little evidence for ongoing neurogenesis past early postnatal human life, there's been a Spanish group that's led some work to refine immunostaining procedures to observe neuroblast-like cells throughout the life of the human, and that these neuroblastic cells are also detected in brains of individuals with Alzheimer's, and that these neuroblast-like cells decline in population over age. And so I noticed one of your early quiescent stem cell markers is ApoE for lipid metabolism. And it's not a coincidence that ApoE is a gene that is important and relevant for certain alleles with regards to Alzheimer's risk and decline. So putting those two together, would you be inclined to write to these investigators to look at these molecular features that you have profiled in your murine stem cells, and to see if those gene expression traits line up in some way, or is there any strong comparative value for the single cell labeling that you've done that can give us some insights as to whether or not we truly have evidence in the adult human brain for these stem cells that can be reactivated and may serve a function that's related to age dependent decline and Alzheimer's cognitive decline?
Yeah, there's a really interesting group that we did a little bit of work with recently. There's a paper coming out where they actually looked at this question. So the first author's name is Charlie Arber, and they were looking at mutations in beta secretase and how that actually also alters notch signaling and accelerated neuronal differentiation. And they were hypothesizing that that could precipitate some of the hippocampal degeneration and stuff that you see in Alzheimer's disease. But yeah, it's fascinating because in some ways it's very much putting the cart before the horse, because people still can't decide whether it actually does exist or not in humans. But the Spanish group, who published that beautiful data last year, provided some really good evidence. I guess the question what everyone is asking is, can people actually find the resident stem cell population? Because no one's shown that yet. These could just be neurons that are taking 20 years to mature, perhaps. So if someone can find the stem cells, that would be really exciting. So, but I have a feeling that that might happen soon. So.
Hello everyone, my name is Jerry and I'm one of the technical application scientists for BioLegend in the Australia and New Zealand region. I'd firstly like to thank the ADNF committee for organising this event and giving BioLegend the opportunity to sponsor. I think this is a wonderful initiative and I'm very much looking forward to all the talks today. So BioLegend is a privately owned global biotech company incorporated in 2002. We're headquartered in San Diego, California, and this is a picture of our new headquarters which opened in mid-2019, along with a picture of our founder and CEO, Jean Lay. Our mission is to enable legendary discovery from research to cure, and we aim to fulfill this mission by developing technologies and manufacturing products across research areas, including but not limited to neuroscience, stem cells, immunology, proteogenomics, and oncology. I'd also like to mention that BioLegend is always interested in forming partnerships with researchers. As an example, we work actively with groups at the Garvan Institutes and also other institutes around the world to test emerging technologies. So if there are any technologies that you'd like to work with us on, or if you would like to discuss anything I talked about today in more detail, please get in touch with me or my colleague Hongyan, or you can check out our website. I've also listed the contact details of our distributors in Australia and New Zealand, and I will stay around in the chat, so please feel free to message me there. Thank you very much.